Okay. Now you can see you are presenting, right? Just let me know whether you can see this name of the chapter. Unmute yourself. Can you see cell structure and its organization?
Yes, ma'am.
Okay. Yes, yes. Now we will begin. Now this is the fifth chapter from which we are going to start with. So the name of the chapter, chapter number 5, that is cell structure and its organization. And we are not going very fast also, because this is your first lecture after a long gap, and as and when you find something different, you can note it down and ask at the end. Okay. Now everybody knows that whenever we want to study cell which is not seen by our naked eyes, so what is required is microscope. And for the first time, when Antony von Leeuwenhoek invented the microscope in late 1600s, which first showed that all living things are composed of cells. Also, he was the first to see microorganisms. And you can see this picture which is there in your textbook also. That is the primary structure of the microscope formed by Antony von Leeuwenhoek, through which he observed something which was larger than the normal, and what he called now afterwards, how the cell, the name cell was given. Cella is the Latin word that means small compartment or compartment like structures. Cella is small room or small compartments. And in 1665, Robert Hooke, first of all, he gave this name. Cells, as you know, is the basic unit of life. And in 1965, sorry, 1655, this Robert Hooke coined the term celluli. And celluli is nothing but the small compartments or box-like structures who, first of all, observe the cork slide under microscope. Okay, and this is also the picture which is seen in your textbook also, how the size varies from 0.1 nanometer to 1 nanometer, 10 nanometer, 100 nanometer, 1 micrometer, 10, 100, and then 1 millimeter. Then which size is visible to our eyes, that you can get to know with the help of this picture. This is what atom, the size is given, which varies here around 0.1 nanometer. Then C60, it's a type of carbon atom which is its size almost 1 nanometer. Then lipids, proteins, then viruses, then organelles of the eukaryotic as well as prokaryotic bacteria, then RBCs size, animal and plant cells, then pollen grain size, human egg, frog egg. So this is what, just for our reference, the size is given. Okay. So this is what you just know, that this is what variation range of the size of different cells, how the cells vary in size and also shape. That will come to that point later. So the cells vary in size. Now everybody knows that there are basically two types of cells: prokaryotic cells and eukaryotic cells. You have learned it to some extent, but right now we will be learning it in little a greater uh, uh, range, or the extent, you can say, in a large more explanation, more explanatory manner we will be studying this. So prokaryotes and eukaryotes are differentiated with a very specific point, that is, with membrane bound nucleus and without membrane bound nucleus. This is a major point with which the cell has been divided into these two categories. That is, prokaryotes or prokaryotic cells, they lack membrane bound nucleus, whereas eukaryotic cells, eukaryotes, they have membrane bound nucleus. Not only nucleus, but so many organelles present in the cells, they show membrane in case of eukaryotes, whereas they lack membrane in case of prokaryotes. So this is the basic difference everybody more needs to understand and remember also. Now, in both prokaryotic as well as eukaryotic cells, a semi-fluid matrix called cytoplasm is present, which occupies the volume of the cell, maximum volume. So the cytoplasm is the main area of cellular activities in both the plant as well as animal cells, as well as both prokaryotic as well as eukaryotic cells. That means whatever the semi-solid, or you can say the fluid, semi-fluid matrix, that main part which is called as that substratum, or that matter, or that matrix is called as cytoplasm, in which different types of organelles are present or suspended. Now, what are these? We will see, discuss later. The one more thing which is common in prokaryotes and eukaryotes is the non-membrane bound organelle, and that is called as ribosomes. So prokaryotic cells lack such membrane bound organelles, but eukaryotics in eukaryotic cells also such few organelles, one of the most important, that is non-membrane bound organelle, found in both prokaryotes as well as eukaryotes is ribosomes. Just why I have told you right now, because for studying, whenever you study, when you study thoroughly, that time it helps you for MCQ especially. So that dash is the organelle which is considered to be non-membrane bound organelle, or dash non-membrane bound organelle is present in both pro and eukaryotes. So that's why I've just given you the example. Okay, beginning with the first type of uh, category of the cells, that is prokaryotic cells. Prokaryotic cells, what they include? They include bacteria, blue-green algae, mycoplasmas, PPLO, that means what? Pleuropneumonia-like organisms. Now, they are very small, and how to differentiate them, that we will see later. Now, these are some of the examples. That is blue-green algae. There are so many blue-green algae where we are going to study them later in detail. Then PPLO, that is pneumonia-like organisms. Prokaryotic cells are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shape and size. So you know that just now I was telling you that there is a range about the size. It isn't it? We discussed just now the range of the size. Just like that, eukaryotic as well as prokaryotic, both the cells, they vary in shape also. Not only size, they vary in shape also. Now in case of prokaryotes, as per the shape, they are known by different names. Now what are these? Four basic shapes of bacteria are observed. Now first one, that is rod shaped structures, you can see, right? These rod shaped bacilli, these are called as, this is bacillus. Rod shaped structure is called as bacillus. Many, they are called as bacilli. Bacilli is the plural, bacillus is the singular. Now sometimes they are present in chains, then they are called as streptobacilli. Okay, they sometimes they are just present in a group, right? Staphylo, like. Then second type is coccus. Coccus means spherical shaped or rounded structures. Bacteria, when they are spherical in shape, then they are called as cocci. Coccus is the singular, cocci is the plural. Now you can see here the chain of spherical shaped bacteria like streptobacilli, they are streptococci. When they are groups, they are called as staphylococci. So coccus means singular, coccus is singular. That is the spherical shaped bacterium, that is called as coccus. Now the third category with the shape you can identify is comma shaped. You can see these having comma shaped structure, so they are known as vibrio. So comma shaped bacteria are known as vibrio. And then when they are in spiral form, when the bacteria show the spiral structure, they are known as spirillum. They are known as spirillum. Okay. When they are in spiral form, the bacteria, they are called as spiruli is the plural, spirillum is the singular. So spiral shaped bacteria are called as spiruli. Again, I will repeat all. Rod shaped bacteria are called as bacilli. Spherical shaped are called as cocci. Comma shaped bacteria are called as vibrio, and spiral shaped bacteria are called as spiruli. Okay. Now, is their range, the organism type, its range. In general, it is given just for information. Okay, that is typical eukaryotic cell. It varies from 10 to 20 micrometer. Then bacterial, typical bacterial cell, it varies from 1 to 2 micrometer. PPLO zero point 0.1. Just now we saw that chart, right? Then viruses 0.02 to zero point 0.2 micrometer. So generally prokaryotes have a cell wall surrounding the cell membrane, and there is no well-defined nucleus. That is a specific feature of prokaryotic cells. And as I told you just now, that the fluid matrix filling the cell is called as cytoplasm. So you remember, in case of prokaryotes, cell wall is present. It surrounds the cell membrane, but the nucleus is not well defined. It is not spatially defined separate nucleus, but nucleus is present, that we generally call it as nucleoid. Now we will be looking, seeing the next slide, then I can show you. This is some of the parts are shown in the prokaryotic cell. One by one we are going to discuss. So genetic material is in the form of nucleus, but this is called as a nucleoid, and this is what the envelope is not seen here. You can't see. This is the genetic material, and it is circular. It is entangled circular form, and this is what the it is not a membrane bound. There is no boundary. There is no membrane around it. Okay, so this is what also called as naked genetic material, but this is generally called as nucleoid. Then along with that, one more point I just want to show you. These names, you can see the name, that is plasmids. Most of the bacterial cells, prokaryotic cells, they show extra genetic material. Like this is the main genetic material which is present in the form of nucleoid, but the same type of self-replicating structures which are circular but with a small size are present. These are small circular DNAs. They are also called as small circular DNAs, or they are called as plasmids because they are having the self-replicating property. Now, what does it mean? Self
Excuse
is,
me, madam. Just
yes,
interrupt you for a minute.
yes.
Just now I have admitted somebody. May I know who it is? Black cat. Who that? Unmute yourself, please. Write your name on that account. Okay, important. Very, all of you remember this. It's very important. Write your name. We don't want any, different kinds of exotic names on that, because it's very difficult for us to recognize you people. So please write your own name on that account. Please change it after the class. Continue, madam.
Yeah. Actually, I was going to tell the same thing in the instructions, but later I forgot as you were going giving the instructions. Afterwards, I also forgot. It's okay. So remember, students, whatever madam has told you. Okay. Now we will again come to this point. So extra chromosomal material, that is having the same properties like chromosome, that is the nucleus. So they are called as plasmids and present in only prokaryotic cells. In most of the bacteria they are present. Now in detail we will study as and when this part will come, but just remember that. Okay. Now membrane extensions into cytoplasm are called as mesosomes. Now whenever, if you see the diagram in your textbook also, the mesosomes you can see in the diagram, if you open the textbook, you can see the diagram also. Here, though it is given as a 3D structure, I will show you few more diagrams which will make you understand easily in the 2D manner. Okay, so membrane extensions into cytoplasm are called as mesosomes, come which extend from the plasma membrane. Just now we were talking about the cell wall. That means the outermost layer is what? Cell wall. Then which in continuation is cell membrane, and that cell membrane extends into the cytoplasm, that is called as mesosomes. Okay, in the next diagram, when it will come, I will explain you again. Okay, so this cell envelope, and in case of prokaryotes, the entire structure, that is called outermost layers, which are considered to be known as cell envelope. So bacterial cells have chemically complex cell envelope, and the cell envelope consists of tightly bound three-layered structure, that is the outermost glycocalyx, followed by the cell wall, and then the plasma membrane. Just now what we were talking, outermost layer as cell wall, and then inside it is the plasma membrane. But in case of prokaryotes, the entire this cell wall is also covered by a outermost layer of glycocalyx. So the three layers together, which are tightly bound, so three layered structure and together is called as cell envelope. Remember, in case of bacterial cells, that is the prokaryotic cells. Now, see, this is what you can see. Remember, outermost as glycocalyx, then cell wall, inside which plasma membrane is present, and all this together, three layered structure is called as cell envelope. Although each layer of the envelope performs distinct function, they act together as a single protective unit. Each layer has its own function, but they are together known as cell envelope. Together it is known as cell envelope, and it has a single protective unit. This is called as. Now, few more points just I want to add in this. That is, bacteria can be classified into two groups based on their chemical composition of the envelope and response to staining. Just now I told you about the envelope, right? Envelope is the outermost layer which contains three layers. Now, this envelope shows a specific type of chemical composition. So, depending on chemical composition of the envelope and response to the staining, these are known as gram positive and gram negative. That staining procedure was developed by the scientist Gram, so the name has come like this. So they are known as gram positive or gram negative based on response to staining. Means bacteria that take up the gram stain, they are called as gram positive, and those do not take up the gram stain, they are called as gram negative. Gram is just the name of the scientist. The stain which is used, basic dyes, the stain that is used, that when the bacteria take that stain, then they are called as positive, that is gram positive, and if they don't take the stain, then they are called as gram negative. So glycocalyx differs in composition and thickness among different bacteria. So remember now, this glycocalyx is the outermost layer, and the entire structure of three-layered structure, that is called as cell envelope. And depending on the chemical composition and the staining, whether they take the stain or do not take the stain, accordingly they are known as gram positive and gram negative. Now, what determines the shape of the cell? Everybody knows that this is the cell wall that determines the shape of the cell. Now you must have understood right now when we were discussing these three layers. The outermost layer, that generally glycocalyx, is responsible not only for the protection but staining method. All together, all three layers together, they are protective in function. But each one has also specific role. Now here cell wall, what is its main role? It determines the shape of the cell. It provides a strong structural support to prevent the bacterium from bursting or collapsing. So shape of the cell is determined by its wall. So cell wall is having its own specific function, and along with that, protective in nature, protective in function. So remember, shape of the cell is determined by cell wall. Now, plasma membrane. Now, the plasma, this membrane structurally similar to that of the eukaryotes. Structurally, it is similar to eukaryotes. Okay, so in detail, we will be studying that cell membrane structure in detail when we will be discussing the eukaryotic type of cell. So now, right now, you remember that all the three layers, that is glycocalyx, cell wall, and plasma membrane together form cell envelope, which is protective in function. Now, coming to the functions of mesosomes, which we were talking, that the extension of the cell membrane, that is what mesosomes. What is the function? That is cell wall formation, then DNA replication and its distribution to daughter cells, helps in respiration, secretes so many substances, that is secretion, then to increase the surface area of plasma membrane in case of absorption, then enzymatic content. Right now you just remember these functions because these are nothing but the extensions of the plasma membrane carrying different functions. As and when it comes, that time again we will discuss. Now, few more points in related to prokaryotes, especially we'll see in some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which contain pigments. Chroma means color. So chroma is the color. So these colorful pigments, they are called as chromatophores, and they are also considered to be, these are the nothing but the membranous extensions seen in the cytoplasm of the prokaryotes, some of the prokaryotes. Okay, so they are called as chromatophores. Now, one more important point which comes with this bacterial cell or prokaryotic cell is flagella. Now, you can see here this extended part of the cell. This is the main body and this is the extension. That extension is called as flagella. So, bacterial cell may be motile or non-motile. Motile means what? It is related to motility, means what? Movement. So, when it is moving, that is called as motile. When it is not moving, then that is called as non-motile. So if motile, they have thin filamentous extensions from their cell wall as called as flagella. So from the cell wall itself, the extension is seen, filamentous extension, that is called as flagella, and that show a range in number and arrangement also. That means what? Sometimes the bacterial cell is having only one flagella at one end. Sometimes the flagella are present, two flagella at the opposite end. Sometimes a group of flagella are present only at one end. Sometimes a group of flagella, tuft of flagella, that means they are present at both the ends. So depending on the type of cell, bacterial cell, there is a range of in the number as well as arrangement of flagella. This is just the simplest form of structure of bacterial cell I have shown, where only one extension at one end, that is called as flagella I have shown, but there are so many examples, other examples where many flagella are present. Flagellum is the singular, flagella is the plural. Okay. The filament is the longest portion and extension extends from the cell surface to the outside. So this is the longest portion outside the cell body. That is nothing but the flagellum. And but along with that, that portion of the bacterial flagellum, now you can remember that such type of extensions which are seen, they are called as the pili because they are made up of specific type of proteins. They are also elongations, elongated structures made up of specific proteins, so they are called as pili. So like filament, like flagellum, this is one more structure that is called as pili. Pilum or pila is the plural. Now here only one at one end, opposite ends, sometimes all over the body they are present, depending on the type of cell. Again, this is this varies. Now, one more extension that is which starts from the cell wall is called as fimbriae. That is small bristle-like fibers sprouting out of the cell. They are called as fimbriae. See why I am telling you, because these are all parts, different types or the parts or the organelles of the prokaryotic cell. Most of the bacteria, different types of bacteria, they show all these different parts. That's why I'm showing you right now. Okay, in some bacteria they are known to help attach the bacteria to rocks in streams and also to the host tissue. Now, just like flagellum, we discussed that motility, movement, that means from one place to another place it can move. That is just like pili. Pili also, they are protective as well as they are helping in the attachment, or sometimes they are helping in the movement. But flagellum more important function is motility. Now here fimbriae, here instead of motility, most of the times how these fimbriae are helpful, they are just like bristles, bristle like fibers present all over the body. In most of the bacteria, so many bacteria, these are the extensions, bristle like structures or fibers present all over the body. Generally their role is to get attached to the host tissue. So with the help of fimbriae, these bacteria get attached to the host tissue. Sometimes when they are aquatic, they whatever the substratum is present, maybe that substratum is in the form of rocks, they get attached to the rocks with the help of fimbriae. Sometimes they get attached to the host tissue, to whichever they are growing in, that also helps there. So here you can see the flagella as well as fimbriae in this diagram. Okay, this is the tuft of flagella, large extensions, this is, and these small bristle like structures, they are called as fimbriae. Okay, now the next that we were talking right now, that is about the ribosome, which is considered to be non-membranous structure present in both pro as well as eukaryotic cells. So the size of the ribosomes in prokaryotes is smaller than eukaryotes. So they that is size which is mentioned is from 15 to 20 nanometer, and their main role is the protein synthesis. Now that you have learned in your previous standards, but still in detail we will be studying how it looks. Now, several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysomes. This terminology you might not have learned previously, so remember that poly means many, ribosomes. At the same, it's is the same terminology for polyribosomes is called as polysomes. Okay, so poly is many, ribosomes. How? Now this is mRNA. You can see this thread-like structures, blue colored structures. Suppose this is mRNA. This is a single ribosome. So like this, it gets attached to mRNA or the single mRNA. Many such ribosomes come close and get attached. So these many, when they get attached on the single mRNA, then that is called a chain of ribosomes on mRNA is called as polyribosomes, or they are called as polysomes. Okay. Now you can see here their role is in the protein synthesis, that we will discuss later at the time of protein synthesis. Now you can see here this part is larger in size and this part is smaller in size, right? So this is called larger subunit and this is called as smaller subunit. In case of prokaryotes, the ribosome type is of 70S type. 70SS represents Svedberg's unit, and that when we will discuss that again, I will show you the diagram in case of eukaryotes. When we will discuss that time, again I will show you the diagram as well as its formation of smaller and larger subunits as 50 and 30, but they are of 70S, they are called as. Now, what are the inclusive bodies or inclusion bodies which are present generally in most of the prokaryotes. So reserve materials in prokaryotes are stored in the form of in the cytoplasm in the form of inclusion bodies. They are called as inclusive bodies or inclusion bodies. Now their role is what? Reserve store material or the storage of the material. And now depending on the type of storage material, they are known as. Okay, so the inclusive bodies are bounded by any membrane system and lie free in the cytoplasm. These lie free in the cytoplasm of prokaryotes. They may be phosphate granules, they may be cyanophysin granules, or they may be, these inclusive bodies might be in the form of glycogen granules. So these are some of the examples of inclusion bodies. That means they may store, these are nothing but the storage house, or they are meant for the storage purpose, temporary storage purpose. So these are in the form of these inclusive bodies, may contain phosphate granules, cyanophysin granules. Cyanophysin, that is a pigment. Okay, I'll tell you. Cyano word is related to blue. Physin, that word is related to the brown, or that cyanophysin is a type of pigment. Okay, granules are present. Glycogen, reserve food material is in the form of glycogen. So this is what meant, these inclusive bodies are meant for the storage purpose. Remember, inclusive bodies or inclusion bodies which are present in the prokaryotic cells, they may contain different types of storage materials in the form of like phosphates, cyanophysin granules, glycogen granules, like that, fat material, etc. So these are some of the examples. Then gas vacuoles are found in blue, green, purple, and green photosynthetic bacteria. Now, there are different types of bacteria, and depending on the color of the pigment they show, they are known as blue-green bacteria, or they are also called as BGA, that means blue-green algae. Then the purple bacteria, because they show such type of pigment. Green photosynthetic bacteria, because their pigment is green. So depending on the color of the pigment they show, they are known as green, blue-green, purple, etc. And they show gas vacuoles also. Now again, I will show this diagram of gas vacuoles, or this one whenever we will be discussing the next part again. Okay. Now, if you have understood, these are some of the basic, common, generally observed organelles present in prokaryotic cell. And whatever the remaining organelles are common present in pro as well as eukaryotic cell, we will be discussing in now eukaryotic cell. Here also a very little part, just some common points or some general points about the eukaryotic cells we will start. uh, Eukaryotic cells, where they are present, now you know, all multicellular maximum, these multicellular organisms, most of the multicellular organisms, they show eukaryotic cells, right? Now what they include? They include protists, plants, animals, fungi. So in eukaryotic cells, there is an extensive compartmentalization of the cytoplasm through the presence of membrane-bound organelles. That is what the basic difference between prokaryotes and eukaryotes. In case of prokaryotes, these membrane-bound organelles are not seen. In eukaryotes, membrane-bound organelles are present in the cytoplasm, so they look like small compartments. So that's why this is called as the cytoplasm shows such type of compartmentalization, because cytoplasm is showing such type of membrane-bound organelles. That's why this is called as that compartmentalization of the eukaryotic cells. Okay. These are some of just examples like paramecium, euglena, then plants, animals, fungi. These are present. These are the examples of eukaryotic cells. So how or what makes the cell eukaryote? That is genetic material. So in eukaryotes, the genetic material is organized into chromosomes. Everybody knows that. Nucleus is well organized, nucleus is very well developed, that is the membrane bound nucleus present in eukaryotes, and the genetic material is in the form of chromosomes. Now again, some are common, like prokaryotes. Eukaryotes also show cell wall, but there are exceptions that where cell wall is present in eukaryotes and where it is not seen, isn't it? No, how and when just we discussed already that Robert Koch observed the cell structure, cell like compartments or cell structures, first of all, but in which cork tissue, that tree, that is oak tree. And this one also we have learned that Anthony von Leeuwenhoek, first of all, observed, and then cell to cell interaction, and the one more thing, that is the barrier, that is the main role of the cell wall. It becomes two ways process. That means one is what? Cell wall, how it helps cell to cell interaction. The cell wall may communicate with the another neighboring cell with the cell wall. That is what cell to cell interaction. And also it acts as a barrier for undesirable macromolecules to enter in the cell. If the cell finds that it is not good for the cell, then that time cell wall works as a barrier. It doesn't allow everything to come inside the cell. So in this way, it works as a barrier. Now, only one about the cell wall we will talk and then we will stop. That is about the plant cell. Now everybody knows that plant cell is having cell wall, but animal cell lacks cell wall. So in general, now we will talk about the plant cell. So plant cell, what it possesses? Cell wall, plastids, and large central vacuole. How these are the points, or these are the things which totally differ from the animal cell. So centrioles are absent in almost all the plant cells, whereas centrioles are present only in animal cells. Cell wall is present only in plant cell, which animal cell lacks it. So plant cell shows the presence of cell walls, plastids, and large central vacuoles, which are absent in animal cells. Vacuoles are present in the animal cell, but they are of small size. And centrioles are present only animal cells, which plant cells do not show. Now, along with these 2-3 points, there are many more points with which we can differentiate plant cells from the animal cells. But at the end of the session, now not this session, at the end of the chapter, we will be discussing all those points. Right now, you just remember that how we can differentiate the plant cell from the animal cell. And now the most common part which we were talking about in case of prokaryotes, also about the cell membrane, which is similar, almost similar. Few basic differences are there, but almost similar to that of the cell membrane which is present in the eukaryotic cell and prokaryotic cell. Now we will just discuss about the cell membrane and we'll stop here. Okay. The detailed structure of the membrane was studied only after the advent of electron microscope in 1950s. The chemical studies on the cell membrane, especially in human RBCs, enabled the scientists to deduce the possible structure of plasma membrane. This is what you have learned. It is there in the textbook also. You have to learn it also. Okay. So what the basic important points about the cell membrane you need to remember is that it is composed of lipids arranged in two layers. Bi means two, layers. So you can see in the diagram, this is one layer of lipid and this is the another layer of lipid. And you can see how you can see these this structure, this layer. This is what rounded structure, spherical structure, that is called as head, and this filamentous structure that is called as tail, right? So you can see that the lipid arrangement in the cell membrane in case of whichever, that means it may be prokaryotic or eukaryotic cell. Here you can remember it is two layered, that is lipids are arranged in two layers, so that's why this is called as bilayered, and they are arranged within the membrane with the polar hydrophilic head. towards the outer side, and the hydrophobic tails towards the inner part. Now, why I have made this red, you can understand. Hydrophobic means which, phobic means which are having fear. Hmm. They are having fear or they hate. Hydro generally word is related to water, so they do not have affinity with the water, whereas philic means affinity with water. Hydrophilic means affinity with water. So heads are having the affinity with water and tails are not having affinity with the water. So in this way they are arranged. So these two lipid bilayers are, this is one layer, this is another layer. Heads are present arranged outwardly, whereas tails are inwardly directed, right? So these are the tails and these are the heads. So this is one layer, this is another layer. Okay, so lipid bilayer, this is called as lipid bilayer. So hydrophilic, heads outwardly present directed, and hydrophobic tails inwardly directed. Now along with that, you can see this purple color structure that is called as protein. Now in this lipid bilayer or plasma membrane layer, not only the lipid bilayer, but in between you can see the proteins also, and how they are known as, that also we are going to see. You can see this structure which is present, all these purple shaped structures, that is what called as, these are all proteins. Now they are known by different names: extrinsic proteins and intrinsic proteins, peripheral proteins. Now when you can see on the along the periphery only, then they are called as peripheral proteins. Okay, when they are present throughout the layer, bilayer, lipid bilayer, they are called as intrinsic. And when they are just superficially, peripherally present, they are peripheral protein or extrinsic proteins. Now, so in your diagram also it is very clearly shown. Say you can see the diagram and you are going to learn it. You can see the diagram on your, in your textbook. You can see the entire structure of, and this is called as fluid mosaic model. Now this is first of all was explained by Singer and Nicholson in 1972, but you can remember this structure that is made up of phospholipid, bilayer and protein. So what the plasma membrane contains? Two layers of lipids and proteins. So this is called as phospholipid bilayer, and you can see this is what these are in between present, which are present throughout the layer. They are intrinsic proteins. Okay, and just seen only at the periphery, if the proteins are present, they are called as peripheral, and the entire bilayer, if it contains, they are totally intrinsic, and just present outwardly, then they are called as extrinsic proteins. So they are just known by the situation where they the where they are present, accordingly they are just known as. So proteins which are intrinsic, they occur at different depths of bilayer, means they may be present. These are all intrinsic proteins which I am showing you here, they are all intrinsic proteins. Okay, and extrinsic proteins, they are just present outwardly. Okay, proteins occur at different layers along the different depths of bilayer of the lipid, phospholipid, they are called as intrinsic proteins. Now so you remember extrinsic and intrinsic proteins, and as per the location they are known as. Okay, now we will continue again. Little part is remaining about this membrane, that is plasma membrane model. First of all, explained by Nicholson and uh, the Singer and Nicholson. That is, this is model is called as fluid mosaic model, which is the most widely accepted or the most accepted model. Okay, this ensures that the non-polar tail of saturated hydrocarbons are protected from the aqueous environment. You can understand the meaning. See, because these tails are not having affinity with the water, they remain protected from the aqueous environment, as they are situated towards, means inwardly they are present, so they are automatically get protected from this aqueous environment. And chemically, lipid components of the membrane mainly consist of phosphoglycerides, phosphate and glycerides. So the membrane also possesses proteins and carbohydrates. Glyceride, glyco, that word is generally related to glyco, that is carbohydrates and proteins. So the membrane of our, and this is what special thing, that is the cell membrane of RBCs, which shows 52% protein and 42% lipids. We are going to just wind up with this structure, integral and peripheral membranes. As I showed you, this is what the integral membrane protein, and this is just peripherally it is present, so the peripheral membrane protein. And this entire structure, the lipid bilayer, phospholipid bilayer, in this diagram you can see, and as it is present throughout, so this is called as integral membrane protein. So integral membrane protein, peripheral membrane protein, that you remember, this is just showed in this way, the one, this and this is one and the same, and when present only on the periphery, they are called as peripheral proteins. Okay, peripheral proteins lie on the surface of the membrane, while integral proteins are partially or totally buried in the membrane. This is what the basic difference. Now we will stop here, because what you need to learn, or what is your homework, I will tell you. Let me stop the presentation and we will discuss